Assalamualaikum warahmatullahi wabarakatuh dan selamat sejahtera kepada semua penonton dan pendengar yang budiman. Selamat datang ke sesi wacana ilmu dalam siri Hard and Honest Talk. Tajuk wacana kali ini adalah The Potential Genotoxicity of mRNA Vaccines. Jangan bimbang kalau anda tidak faham perkataan saintifik di dalam tajuk ini. InsyaAllah tetamu kita pada malam ini akan menerangkan apakah yang dimaksudkan dengan genotoxicity. Jadi malam ini saya sebagai host, nama saya adalah Abdul Karim alias, adalah host dan akan bertindak sebagai pemudah cara sesi wacana malam ini. Dan saya telah menjemput untuk kali kedua tetamu untuk kita membicarakan tajuk yang sangat penting ini, Dr Manimalar Selvi Naicker, konsultan Histopathologist dan Statistician. Selamat datang Dr. Mala.
Thank you Prof. Good evening.
Terima kasih kerana sudi lagi menerima jemputan saya kali ini untuk kali yang kedua. Jadi tuan-tuan dan puan yang budiman, sekali lagi saya ingin mengatakan atau menegaskan bahawa wacana yang dijalankan ini adalah wacana ilmiah. Ini bukan suatu perbahasan untuk mencari salah silap atau menuding jari, tapi ini adalah usaha yang berterusan untuk memberikan satu pandangan yang lain ataupun pandangan sisi kepada orang ramai kerana ini adalah isu kesihatan yang penting. Jadi pada malam ini kita akan membicarakan tajuk The Potential Genotoxicity of mRNA Vaccines dengan Dr. Manimalar, dan untuk membicarakan tajuk ini kita akan jalankan perbualan ini di dalam bahasa Inggeris, tetapi saya akan menyediakan subtitle di dalam bahasa Malaysia dan tuan-tuan dan puan bolehlah turn on the closed caption di dalam YouTube untuk mendapatkan subtitle di dalam bahasa Malaysia ataupun di dalam bahasa Inggeris juga, ya. Baiklah, Dr. Manimalar, sekali lagi selamat datang ke dalam sesi wacana pada malam ini. Jadi as usual, I think we will start this conversation with very basic about the vaccines, because I think the public need to be informed the different types of vaccine that we are having in Malaysia. But there are at least 3 or 4 different types of vaccines being used worldwide. But in Malaysia, I think the most widely used or the popular one is the Pfizer vaccine, which is based on mRNA. And of course, we have also the adenovirus based vaccines such as AstraZeneca, I think. And we also have Sinovac from China. But tonight, I think our focus is to talk about mRNA vaccine because it is being used widespread use around the world. And the issue of safety, especially so-called genotoxicity of mRNA, because now with more and more studies being carried out around the world, we get more data now showing that there's a so-called potential genotoxicity of mRNA. But before that, Dr. Mala, can you just briefly tell us or explain to us what is actually the mRNA vaccine and how does it work? Silakan.
Okay. Yeah. Basically, the mRNA vaccine is they just take part of the genetic information of the virus, right? And they just package it in a lipid envelope. So that is meant to be able to help deliver the virus into the cell. And because the genetic information is already in an RNA code, then in our cell acts as a factory and just creates the protein, and then this protein is then expressed on the cell, and then the body recognizes it as the COVID-19, and the body creates an immune response to it. So when we have an immune response, you have the most immediate immune response, which is your antibody levels, and then you have got the more permanent response, which is your memory cells. So after a while, the antibody levels will go down, and they should go down, because otherwise, our blood will become very viscous. So then that will create more problems. So after a while, the antibody level will go down, and then we have the memory cells, and then next in 2 or 3 weeks time, when we go jalan we go, and then we meet somebody with Omicron or whatever, so we will get the infection, but within 1 or 2 days before the infection can become clinically apparent, our memory cells will produce more antibodies and take care of that infection. So even though we might test positive, but we usually don't have symptoms, or even if we have symptoms also, it's very mild symptoms. So the idea behind the mRNA vaccine is very good. Now, the reason why they chose this type of vaccine is because it's an artificial, it's a synthetic substance, and it's easy to produce. Unlike, let's say, the Sinovac, which is an inactivated vaccine, which means you have to grow the virus and then further process the virus and all that. So this is an elegant way of inducing immunity in our body. But the problem with the mRNA technology is the technology is quite old. It has been around for some time. In fact, even prior to 2018, there were at least 4 or 5 pharmaceutical companies which did try to make mRNA vaccine for things like Zika virus, rabies, and even influenza. Moderna actually started their influenza study sometime in 2015. But the thing is, they never managed to commercialize. It never managed to progress to a phase 3 or to become a commercial vaccine.
Why?
So that's why to many of us, especially those lab medicine physicians like myself, so that sort of rang a bit of alarm bells for us.
Yes.
Because see, influenza is something like COVID-19, thousands of cases every year. So many hospitalizations, deaths. So it's very easy to recruit. So in Moderna at that time, they actually went into phase 1, phase 2 already. So they can recruit patients, but somehow the studies cannot progress. You see, so when we see that, then being lab medicine physicians is already like an alarm bell for us. What is the problem? Why the studies cannot progress in spite of a lot of potential patients? It's not
Big
like
question
rabies.
mark, Big question mark.
Yes, it's not like rabies. You see, rabies is it may be more difficult to recruit patients for a rabies vaccine because who gets rabies. But for influenza, it should have been a simple matter. So that became a very big question mark. So that is why I started looking into the mRNA vaccine actually. Then when the vaccine came out, the product insert of the vaccine, Prof, I show you. I share screen here.
So this is you're talking about the Pfizer product insert, right?
Pfizer, yes.
Okay.
Dr.
As you can see here.
Yeah, you can see the
Yes.
document. So what is this actually a product insert?
This product insert is the piece of document which comes with each Pfizer vaccination. By right, this is supposed to be given to the patient, but because we share the vaccine, normally mass vaccination, we don't give it out, I suppose.
Yeah.
But the information here is meant to be given to the patient. Right?
But we don't get it.
No. Nowadays it's a sort of online and all that.
Okay.
Yeah, this is the patient information sheet, and for the doctor as well. So here it says very clearly, Comirnaty, that is Pfizer, has not been evaluated for the potential to cause carcinogenicity, genotoxicity, or impairment of male fertility. In a developmental toxicity study in rats with Comirnaty, there were no vaccine-related effects on female fertility. Right? So it has not been studied. So when we went to look and see why it has not been studied, this is the European Medical Agency document. So they say here no genotoxicity studies have been provided. This is acceptable as the components of the vaccine formulations are lipids and RNA, and that are not expected to have genotoxic potential. So as you can see,
we lost the document.
Yeah, I stopped sharing.
Oh, you stopped sharing. Okay.
So did you manage to see that?
Yeah, we saw the document just now.
So basically, they just they just assume that an mRNA component cannot cause genotoxicity. So they did not do the genotoxicity studies.
Well, this is puzzling. I think I want to dwell on that a little bit.
Yeah.
And EMA, the European Medical,
Yes. It is the counterpart of the
yeah,
United States
yeah, it's
FDA.
equivalent to FDA, right?
Yes.
They said okay, now the mRNA is actually is being encapsulated in the lipid nanoparticles as a carrier.
Yes.
And they said, okay, the lipid does not cause genotoxicity. But what about the mRNA itself?
Yes, that also they say that it is not expected to cause. This one I believe is because in 2005 there is a WHO document which says that if you don't think that something causes genotoxicity for vaccines, you don't need to test it, right? So yeah, it's a very odd sort of situation for them not to test a brand new vaccine. I think that document was meant for those old vaccines so that
Mhm.
poorer countries can manufacture vaccines. They don't have to go through the cost of all this. I think because it's a very old document, but they have applied it for a brand new vaccine which has never been commercialized as an
Hmm.
excuse not to do the genotoxicity study. So that is what I found quite strange. You see,
So,
this
so,
vaccine is too new for us to make that assumption.
so as you just now you show the product insert, the Pfizer product insert, it clearly said that they have not yet done the what do you call this, the proper genotoxic study.
Mm.
So meaning there is no proper what do you call this, animal studies involving the animal studies and so on.
Basically, genotoxic studies are any kind of studies
Mm-hmm.
where you look at whether your DNA, sorry, your genome can be
Mm-hmm.
affected by the substance. It can be chemical, physical. It doesn't matter. So,
Mm.
does it interact with your genome or not? One
Okay.
kind of genotoxic study is the carcinogenicity, where it causes carcinomas. But genotoxicity can also include other things like autoimmune disease and all that. So normally
So
it is a mix of in vitro, in vivo, and if they can do it on a small sample of humans also. Sometimes it's just a matter of following up patients for
mm,
some time. So I mean the way they do it for different compounds may be different.
yeah. So in other words, when we say genotoxicity, we are concerned about whether whatever we take, whatever vaccine or yeah, vaccine in this case, it doesn't cause any change on our DNA structure, right?
Yeah, it doesn't interact with our
Doesn't interact. So
genome
that is what
toxicity.
genotoxicity means. Okay.
And the
Now,
traditional vaccines actually don't interact
uh,
because they
but
are like
it obviously Pfizer didn't do that genotoxicity
yes,
study on the mRNA vaccine that now
yes,
we are using now
yes,
all over the world, not only in Malaysia, right?
yes, yes.
So I remember in the early part last year when the first phase of vaccination, there were uh, people saying that, or the Pfizer vaccine, mRNA vaccine can cause DNA structure to change and so on. So uh, immediately, I think the ministry and so on uh, basically say no. It's impossible. And when I look at the YouTube video and so on, expert from different medical doctors and say that no way the DNA structure can be changed. But I think now we want to talk about the recent study, the most recent studies by the Swedish group. And this actually, maybe I just share the slides and later on maybe Dr. Mala can talk about it.
Yeah.
Which share the slide, the PowerPoint here. Um, can you see the PowerPoint?
I can, Prof.
You can, eh? Okay. So this is actually the uh, paper published in Current Issues in Molecular Biology by
Mm-hmm.
a group of Swedish researchers from Lund Lund University. So Lund
Yes.
University, I think a lot of people know that is one of the oldest institution in Europe and very highly respectable. So this group is also, very credible. So basically, what they say, the Swedish study demonstrates for the first time the presence of Pfizer BioNTech vaccine DNA in the cell. The vaccine causes intracellular reverse transcription of BNT162b2 mRNA. So sounds very strange, very
That's
alien to
Pfizer.
us. Yeah, but you can help us to explain this one. Transcription of this into human DNA in vitro. Okay. Later on, Dr. Mala can explain also what does it mean by in vitro. It is reverse transcribed by line 1 proteins of the cell. They are not sure if, so this is very important to understand. That's why we say potential. They are not sure if it gets integrated in our DNA or not. Renewing concerns that vaccines may introduce spike protein into the nuclei of cell. Okay. And this one we take from the paper. They said our study shows that this BNT162b2 can be reverse transcribed to DNA in liver cell line HUS7, and this may give rise to the concern if BNT162b2 derived DNA may be integrated into the host genome and affect the integrity of genomic DNA, which may potentially mediate genotoxic side effects. So maybe I think Dr. Mala can you elaborate on this one?
Okay. I think I'll first explain what in vitro means.
Yeah, please. We don't
Okay.
understand.
So when we talk about studying things pathophysiology in in diseases, one we have the in vitro, that's a cell system. Then the other one we have small animal models. Then we have like larger animals, and then we have humans. So normally when a drug or whatever goes through developmental stages, it will go through most of these stages. Now, an in vitro system basically it provides proof of concept or proof of principle. So what it tells us is because the cell system that we use is a human cell system. Here they have used a liver carcinoma system, right?
Mhm.
And the reason that they use carcinoma cells is because it's easy to propagate carcinoma cells. They keep dividing. So when something happens in that cell system, basically it tells us that our cells have the tools to do what it has done,
Mhm.
right? So like now they want to show the mRNA can become DNA. We first must be able to reverse transcriptase, right? Then it can become DNA. If you want to show that it is integrated into the genome, it must be able to splice, and then it must be able to have integrase to integrate, right? So when you do this in vitro studies, it merely shows that our cells have the tools in hand to do all these things. Now, whether
Mhm.
it does it in real life in a living person or whether it does it often is something else.
Yeah.
But we know it can happen. So as you said, Prof, it's the potential. Yeah, that means it's armed and ready. It can do it, right?
So yeah, this shown in the test tube lah. In other words, right?
Ah,
In
with
vitro.
yeah, all the tools are there in our
Yeah,
cells to do
but
it.
it has yet to be tested in a human being. But we are, millions of people already taken the vaccines.
Yeah.
So meaning that, and now from this in vitro study, it has been shown that it is possible
Yes.
that this somehow the vaccine, the mRNA, in a layman language, can cause a structural change in the DNA. Am I right?
Um, so far, we have not shown that it affects the sequence of our genome yet. What the
Okay.
Swedish study has shown that it can reverse transcribe into DNA. But the issue is we were told that it has no potential to do that. That is why they didn't do the genotoxicity study.
Mm-hmm.
You see, so the genotoxicity studies were not done based on what now appears to be an incorrect assumption that it cannot reverse transcribe.
Okay, to put it simply, what would be the implication on long term for those who have taken this mRNA vaccine? Should we be worried?
Okay, if you want to ask, I mean, for me, I would not be particularly worried, but I would
Mhm.
want to inform the patients of it.
Yeah.
So for me, the concern is not so much how much of genotoxicity, but for me, it's mainly that we should put it in the informed consent. That you see, we have not done the genotoxicity studies. So any potential risk we don't know, because now at this point in time we do not know what the risks are.
Mhm.
Yeah.
So if that is the case, there are still a lot of uncertainties, right?
Yes.
In terms of the so-called genotoxicity and what it can do in the long term and how it would be manifested in our body, right?
Yes.
So if that's the case, the patient or not the patient, I mean, the people, those who want to take the vaccine must be informed of this, right?
Yes.
And they provide all the possibilities which we have not yet not very certain about it.
Mm.
But this is not done, and I think people are keep being told that take the vaccine, take the booster, but not being informed of the so-called the risk, the potential risk.
Yes.
So to me, that is not really balanced in terms of the benefits and the risk. People are being told and informed about the benefits only, but they don't mention about the risk.
Correct.
That's not right, right?
Yes, so that was my question to the
Mm.
government. As you know, because we gave the patients the vaccine without doing the genotoxicity study,
Mm.
trusting that what the experts told us that it cannot enter it cannot reverse transcribe into DNA,
Yeah.
uh, is now not true,
Mm.
right?
Mm.
So what they are saying is they have not proved that it can fuse with our own genome, but that is not much. It's not very comforting because they have not proved that it doesn't fuse with the genome either. They don't know. You
Mm.
see, so basically there is unnecessary uncertainty, and this should not have occurred in the first place. And now that we have no genotoxicity studies, I think we should inform the patients of this. It is a
So,
possible risk.
in other words, we are not out of the wood, so to speak.
Yes,
Tak
because
selamat
in
lagi.
medicine, the absence of evidence doesn't is not the evidence of absence. Just
Ah,
because we so far do not know of any incidents of genotoxicity doesn't mean that there is no genotoxicity. Maybe we haven't looked hard enough. In fact, in the
okay.
case of this, we have not looked at all because we haven't done those studies.
Yeah,
You see?
yeah. You mentioned about. It's interesting when you mentioned about. Let me find the words that you use. Let me share the screen now. Uh, yes, 1 second. Yeah, can you see this?
Yes.
This is your words. I, I take this from the Facebook. Medicine is not criminal law. No, that's the other one actually. Yeah, when you say the absence of evidence does not mean this one is different. I think
Yes.
this one is different. So can you repeat again? You said the absence of.
The absence of evidence.
Uh, oops. Hello.
Prof.
Oh, sorry. I know what happened. I. Okay.
I can send that absence of evidence thing to you, then you can put it in the thing.
Okay.
Ah,
Uh, well, anyway, never mind. We can come back to that.
yeah.
Let's talk about the other thing. I think there's another paper before the paper published by the Swedish group, right? Um, and that is. Do you want to share that paper, or do you have it?
Yeah, I'll share that, Prof.
Okay, and
Okay.
maybe we can talk about it because I think there's another important thing that we want to understand.
Yes.
Um, when we get
Dr.
when we do the
Manimalar
testing.
Selvi Naicker joined.
Okay, yeah, it's the one, right?
Yes.
Or this is the paper by the Swedish group. No,
No, this is the
oh,
paper
it's a different
by
one.
MIT. This
Mm-hmm.
paper came out in July of 2021.
Mm-hmm.
So this was actually the seminal paper. So this paper was done because they were wondering why is it patients who had recovered many months ago from COVID-19 infection,
Mm-hmm.
how is it their body was acting like a factory and producing viral parts? Of course, these viral bits were not infectious, but
Mm-hmm.
they were still viral bits. So
Mm-hmm.
they postulated that there was integration into of the viral infection into our human genome, and this was producing these viral bits, right? So
When,
this,
when you say the viral bits, you mean is that the spike protein on the virus, or is it any part of the virus?
any part of the
Any
virus.
parts of the virus?
Yeah, but the one that we were detecting was, of course, the spike protein because our tests were detecting that.
Mm-hmm.
Yeah, so the job was made a bit easier for them. So they were wondering why are people testing positive about few months after after their infection, but they were not infecting anybody else. So it was
Yeah.
like empty virus being produced. So the reasoning was that the body is now acting as a factory to produce these bits. So they designed a study to show
Mm-hmm.
that the live virus, the C19 infection, can infect our cells, can reverse transcribe and integrate with our genome, and then express RNA and produce the viral parts, right? And this paper is very important, Prof, because uh, it pinpoints that key tool which can do all these things, and that is what is known as the line retroposons. You see, for when we study genetics, we have to understand that a lot of things that we studied in the past are not correct because initially when we studied genetics, it was the central dogma that DNA becomes RNA becomes protein, right?
Mm-hmm.
Then suddenly we were informed that there's something called reverse transcriptase. RNA can become DNA, and then we thought that human beings don't have reverse transcriptase. Then we find out human beings do have reverse transcriptase, but only
Mm-hmm.
thing is it's not very active. So the reverse transcriptase that we have is known as line 1, right? And this is a very special kind of reverse transcriptase. It's called a retrotransposon because it can do a lot of things. It can act as a reverse transcriptase. It can also cleave into into our genome, and it can act as an integrase. So basically, it's an all-in-all kind of a tool. So this line and sign and this retroposon things can basically they are a complete toolkit to be able to integrate RNA into our genome. So that this paper is very important because this tells us that it is possible that the Swedish, if they had continued with their experiment to show integration, that a mechanism already exists.
Mm.
Yeah, so that is why this paper is very important, and it's a very elegantly done paper because every step of the way, for every step, they repeat it 2 or 3 times using various kinds of tools. They don't just do it each experiment once and no. Like you see, the integration, they have used 3 different methods.
Mm.
So one confirms the other kind of thing. And this is a very special study because it also shows that uh, the protein can whatever is integrated into our genome can also express, and here they have used what is known as our bronchoalveolar lavage. We know that a lot of these COVID patients, they are in ICU, so they have a lot of this lavage means that their lungs are being cleaned out, and then they take the cells from that cleaned out fluid and then they have it sequenced. So they are able to show that those cells also have this RNA integrated into those cells. So this is a very elegant experiment. Of course, a lot of people didn't like it. It was very severely attacked. But at the end of the day, the data stands. So, I think we can conclude from this paper that human beings, we have the tools to integrate RNA into our genome. So that is why I thought that this paper is very important. And this was the precursor paper. So of course, naturally, when we know that the virus, the live virus can do that, the next question is, can the RNA of the vaccine do the same thing? So that is when the Swedish paper comes into play. So the Swedish answered that question. So basically what they did was, okay, this is an extremely elegant paper as well. They used liver cancer cells, and they basically bathed liver cancer cells with the RNA vaccine at various concentrations for various hours. And then they were able to show here that the what do you call it, that the vaccine has entered into the cell, and the vaccine also, so the postulated mechanism or the toolkit was actually the line 1 gene, right? So here they were able to show, like here, that there was upregulation of this gene, right? And what they basically showed in this is the RNA, mRNA can go and disturb the gene, right? So that is what they basically showed here. And then this is that RNA, that line 1 can now expressed as a protein. So this is that part, right? This is known as immunohistochemistry dual staining of protein, right? We do immunohistochemistry of protein, and here also we can see that protein is also increased. So these two diagrams basically show us that somehow this line 1 has increased, right, in expression as well as protein formation. Then what they did was, then they went on to look for, okay, all these things are expressed fine, but can we get DNA which was reverse transcribed from the RNA, right? So this is the part where it gets quite elegant, in the sense that because here you would use a PCR to look for DNA, right? So if you don't have DNA, the PCR is not going to show up anything. So here they managed to show that there were some PCR products. That means there was DNA produced. Now, whether that DNA is our own DNA or DNA from the virus, we are not sure yet. But we can see that, it matches the weight of the mRNA, component. And then they go and sequence that DNA which they got, they go and sequence it, and it's a perfect match with the RNA, with the mRNA vaccine, right? So this is known as Sanger sequencing of, they just call it an amplicon. That means they don't know where it's from, but it is a perfect sequence of the mRNA vaccine. So basically what this shows is that mRNA vaccine can now be turned into DNA. Now, whether it is from an integrated into the genome, all that we don't know, but it can become DNA. And the interesting part is, one of the news media outlets contacted Pfizer, and Pfizer has given, apparently has given a remark where they just say that, oh, the genome will not be altered, right? But they didn't dispute the study, or didn't dispute the fact that mRNA vaccine can become DNA. So once this happens, this ability is there, and we know that this line 1 by itself has got the ability to cleave and integrate DNA into our human genome. So then, without the genotoxicity studies, things become very uncertain. Now we don't know, is this going to have an effect on us or not, right? So this is, as I said, we are not in a very nice position now without the genotoxicity studies. So as a doctor, now I'll be very scared to go and tell patients, don't worry, nothing will happen, your genome is not affected, it cannot become DNA. And the thing is, it can go and disturb your line. See, they have proved that it can go and disturb your line gene, because that gene is now overexpressing in some of those things. And all these things were not supposed to happen with this vaccine, you see.
That's
It
the
was just
thing.
supposed to be in the nucleus, produce some protein, and go off, and that's it.
So we can say that there's a lot of things. They are still very uncertain. A lot of
Yes.
work in progress. The scientists are looking at it, especially the independent scientists, not those sponsored by the big pharma. Um, and yet, I think we have been. I mean, we have been told that go ahead and take the vaccine. It is safe. It is safe. There's not even some explanation on the possible risks. They just downplay everything about the risk. That's what my impression is. What's happening now, and to me, that is not the right thing to do. Right?
Correct, Prof. I agree with you, because I think in medicine is very clear. When there is a material risk, however small or however remote, the
Mm,
patient should be informed. Because I would think that many of older patients, they wouldn't mind taking that kind of risk, but a
mm.
lot of young people would not want
Yes,
to take that. Yeah.
the young people, those teenagers, those
Mhm.
young, children, they have a long way to go in their life, and
Yes.
I cannot imagine, if any of these studies really turn in, I mean, it become real, meaning that it is manifested in our body, and it turn into, I mean, it create havocs in our body. Can you imagine this? The young people, they have 30, 40, 50 years ahead of them, and they will have to live their life having to, and suffer all these bad long-term side effects that we don't know, because it has not yet manifested. And yet, I think some of the what I have been hearing from the medical doctors and medical professional, that they say, "Oh no, it's been 2 years and we have not seen any uh, side effects, except for some small number of serious AEFI." And to me, I simply cannot accept that kind of words, because we do not know yet. Yeah. Okay. And you mentioned about the earlier paper that even after 2 months, 3 months, if we do the, because I think the doctors have been saying that after you get COVID, and after you have tested positive after a few 7 days or so, don't do PCR, because the PCR
Yes.
can still show positive results after up to 90 days. And you mentioned in the earlier paper that that's the case because of the bits of the virus, right?
Yeah.
Uh, but how that is, what is the kind of the implication of that? Is that something that you should be worried about?
Okay. This is just the way I look at it, Prof, because they use
Mm-hmm.
bronchoalveolar fluid. You see, the virus is a respiratory virus, right?
Mm.
It goes in through our respiratory system and fuses with our respiratory cells. So the good thing is our respiratory system and the cells there are not permanent cells, which means they keep renewing, right? So chances are those layers which are older will just shed, and new layers will come. So that's probably the reason why that after a few months, when the old layers have all shed, we stop producing what do you call it, this viral particles anymore. There's a renewal. But you see, we have other kinds of cells in our body which are what they call stable cells. That means they can reproduce, but they don't reproduce all the time. And then we have got some permanent cells which we cannot change. They
Mm-hmm.
don't shed at all. So the implication, if the virus goes and starts sitting in, the reproduction capability is there in um, one of these kind of organs, then we are worried about it lah. But I think for the for the infection itself is basically the respiratory system.
Okay. Okay, maybe the last part of our discussion in this session. Maybe I want to ask you about I've seen a report showing that the spike protein might have been found in some of the our organs, like in the spleen, in the liver, in the kidney. I remember there quite a long list, and it is present a very high concentration, uh, quite significant amount in, can't remember, but liver is, and spleen. I think spleen is one of the. So have you seen that report, and what would be the implication if this, and how, where this spike protein come from? Because my understanding in the spike protein will be kind of destroyed after he has done the job. Can you explain a little bit on this one?
Okay, Prof. See, the problem is, again, we were told that when you give the injection, it's only in the deltoid muscle.
Mm.
It's not a traveling vaccination.
Mm.
But the rat studies that they have done, the mice studies, actually show that about 20% goes to the liver and to other organs as well. So I am not sure why they told us that it doesn't travel anywhere except maybe to the regional lymph nodes, when the small animal study shows that it does travel, right?
Mm.
So the thing is, the next question is, what travels? Is it the entire vaccine which travels, which means that now it can go to your liver and then make a home there, or is it just the protein which will travel? Because the protein, we know that it does travel.
Mm.
It's in circulation. That's the antigen, right? So uh, the bio, from what I understand from the documentation, they have not actually done a formal biodistribution study.
Mm.
So what they have done is what they call as proxy biodistribution study, and uh, and what they've done in the rats show that it moves around.
Mm.
So that implication is also not very clear to me. So that's the thing I was asking. That the government should make all this very clear to us. Not and the other thing, Prof, which I found very difficult was, you see, very early on they started demonizing experts in the field. You see, like Prof. Robert Malone and
Yeah,
all that.
McCullough, I think is that.
Yeah, and then the pathologists and all
Yeah.
that, when they started speaking out, I don't know how they got so demonized in the media. And
Mm.
for me, it was quite irritating, because these are people with the right degrees and credentials.
Yes.
We may not like what may not like them, but what
Yeah.
they say counts,
Yes.
because they have the right degree, right? And so they were sort of driven off the scene. So it's very difficult now to collate that information. I know that in Germany, one of the pathologists there has done a series about a few cases to show
Mm.
this spike protein toxicity is causing endothelialitis, and and the ramifications of it. But we are not able to sort of codify it nicely and have a look at it. You see, because these people have just been driven off the screen. Very irresponsible behavior,
Yeah.
considering that they have got all the right degrees. Why would you not want to listen to them?
I think the mainstream media, the mainstream social media as well, I think they are complicit in silencing the experts, the real expert in the field, like Robert Malone, uh, McCullough, I think. They are very vocal. They don't have any vested interest. They are the real expert in the area, but
Yes.
somehow people just, I mean, the media just silence him. And I don't know about our government, our experts, whether they are also referring, his their work is listening to them. I don't know,
And
because
it would have been nice if they had our government had invited him,
yeah,
and listened to that to them. Because I'm sure even the government knows that definitely drug companies, they spend a lot of money on advertising, so
Yeah.
there will be negative advertisement against those people who, speak against them. But it's definitely the duty, to me as a doctor, I want to listen to experts,
Yeah.
people who have got the right degree, right qualification, right?
Oh, the thing is, we want to have a balanced view. We cannot just listen from one side. We have
Correct.
to also listen from both sides. Here are the experts, and here are the probably. So that we have the exponent and the proponents
Mm.
of vaccine. So let's thresh out. Let's put the evidence on the table and look at the evidence and argue and debate on the evidence rather than simply on the emotion or uh, everything has to be guided by the evidence. Look at the evidence, what the evidence says, and let the figure speaks. But I think it's not really happening that way. The strong view from the other group like Robert Malone and so on, they just simply dismiss. They don't want to listen to these experts. So there's something that I really very puzzling, and I don't really understand why. So I
Correct.
think,
Even for
yeah, this is the reason why we're having this conversation, because we want to bring the other side. Let's talk about it, and look at the data, what it says, and so on, and just understand that there are still unfinished work here, that we need to be cautious, always be cautious, and we decide now whether whatever that we take, these are the risks. But the risk has not been well informed to us, to
It's
the,
not
to the
only that, Prof. Even for doctors also, like I've been asking, what is the protocol for an autopsy? See, when you want to do an autopsy for, a
yeah,
vaccine case,
yeah,
you need a proper protocol so that you don't miss anything.
mm,
Like, okay, from the heart, where do you want to take the sections from? How many sections you want to take? The
mm,
minimum, what kind of stains that you want to do? And all this knowledge comes when you listen to the experts.
mm,
So when they tell you, okay, what kind of pathology are they looking at? What is the pathophysiology? So based on that, they will tell you, okay, do this thing. Then only you, because if you don't do those things, you're not going to find anything.
yeah.
You will be forever in the dark. So that
But
is what I was saying.
if you purposely don't want to find it, then you
That
don't.
is very puzzling to me. I mean, how come after 1 year as a pathologist, I am not able to access a postmortem protocol?
Yeah,
I also don't know now for heart, how many sections should I take? What stain should I do? Right?
yeah,
And how are the pathologists, the forensic pathologists doing the postmortems? And
mm,
who is advising? Who is so everything is a question mark. And the problem is what the mind doesn't know, the eyes will not see.
yeah.
So if you don't know what the pathophysiology and the mechanism is, how will you know what stains to do?
So I'm just wondering, after, I mean, when we look at the report, CDC, what do you call this? VAERS, VAERS. Sorry,
There's
VAERS. The reporting system, voluntary adverse effect, I think, reporting system by CDC. Uh, Clearly, there are reports of what do you call this, fatality, right?
yes.
Uh, uh, and they actually relate that to COVID as well, and there are adverse effect and so on. So meaning that there are death cases due to COVID. But in Malaysia, we have yet to have a single report on death related to COVID. So as the layman, we are just wondering whether, as you said just now, whether there's a proper protocol to really find out the root cause and the causality, whether this is really caused by vaccine or not, and we don't know, right? We don't know. Um, all right, so I think probably, do you have anything to add to our discussion tonight? Maybe
No, for me the main thing is, Prof, is it's the consent. I'm very concerned that young people especially are aware.
Mhm.
You know, Like, I'm very pro giving this vaccine to older people. I myself would go around persuading, but when it comes to young people, you take it if you want to,
Yeah,
right?
yeah.
But you be aware that these tests have not been done.
Mm-hmm.
And these tests are most likely to have greater impact on a young person.
Mm.
Probably genotoxicity is probably not a very important issue for an old person, but for a young person, it probably does carry some significance, right?
Yeah.
So be aware that this study, the genotoxicity study, hasn't been done, and be informed when you give your consent.
All right. So I think maybe for this session, that's about it. Maybe we can have other session to talk about other issues as well. I would like to thank Dr. Mala for uh, sharing your thought and sharing your knowledge, your expertise on the potential genotoxicity of mRNA vaccine. I think the conclusion of our discussion tonight, the bottom line is there are still a lot of uncertain things that we do not know yet on the long-term effect of mRNA vaccine. There are a lot of studies that need to be done, which I suppose should have been done as part of the uh, safety studies, clinical studies, before the vaccine is approved by FDA. But now we know that Pfizer obviously has said very clearly there is no genotoxicity study done, and but millions of people have already received the vaccine, and I cannot imagine, 5 years down the road, what will be the effect at that time. It will be manifested in different ways, but after some time, maybe people forget that this is probably due to the vaccine. So we do not know yet. We do not know yet, and we don't want to be among those. We don't people accuse that we are doing a fear mongering here, or to try to make people, feel feel um, kind of scared or fear of. so um, so once again, thank you very much, Dr. Mala. I
Thank you,
really
Prof.
appreciate your time. I hope what I was sharing tonight can give another perspective, another side of the story to Malaysian people, especially, so that they are aware and they can find out kind of find out more. They don't just take it from us. We are just kind of uh, we are trying to give another perspective. That's all, and let us explore together. Fikir-fikirkan, timbang-timbangkan, think about it, do your own due diligence, study and read, so that we are well informed. We become well informed uh, consumers. All right. So with that,
Thank you
thank
so
you very
much, Prof.
much, and see you again
See you,
in
Prof.
another program. Thank you, Dr. Mala.
Thank you, Prof.